So the nucleus is the organelle that actually stores DNA. So DNA is the genetic information, right? So encodes for the genetic information. We mentioned before that we oftentimes think of DNA as kind of the blueprint of all that genetic information, and it's stored inside the nucleus in eukaryotic cells. And so there are many different types of organelles in general within the eukaryotic cells. So we have nucleus and ribosome. So here's our nucleus right here, and you see all these little teeny dots that we kind of see here and here, those are our ribosome. And so the ribosome will attach, we have free-floating ribosomes within the cell, but we also have these ribosomes that are attaching onto the endomembrane system, specifically the endoplasmic reticulum, to kind of allow for sites of protein synthesis. So that's where we actually synthesize our protein. We also synthesize protein that are on the ribosomes that are free-floating within a cell. Additionally, another organelle that we're going to kind of look at in a little bit more details are the organelles that make up the endomembrane system. So that's pretty much this whole system right here, and it's pretty much containing pretty much all the ones here. So we'll look at them in a little more detail there, but all of these are different organelles that are in the endomembrane system, and they help form different molecules that we need for metabolism. We also have energy-related organelles, as we mentioned, which would be like the mitochondria right here. And then we have the cytoskeletons, which is right here. And so these are all the different structures right here that kind of gives some structural support to the cells. So we'll look at those in a little bit more detail. So let's kind of look at the nucleus again in a little more detail here. So the nucleus, as we mentioned, is what stores the DNA. So inside this organelle, we have all of this DNA in here, and we want to protect the DNA from potentially damaging reactions to cytoplasm. Because within the cytoplasm, we have a lot of different organelles that have different enzymes, such as digestive enzyme that can kind of damage the DNA. So we want to store the DNA in this structure right here to kind of prevent it from getting damaged. What separates them is this nuclear envelope. So this right here is our nuclear envelope, and the nuclear envelope is a double layer membrane, so it's also double layer, and it separates the nucleus from the cytoplasm, and it also determines what gets to go in and out of the nucleus. So you can see that within the nuclear pore, we have a lot of these different, nuclear envelope, we have a lot of these pores. So look at those in a little more details. So here's the nuclear envelope, has these different pores, right? And this is another pore right here, and so on. And so these pores allow certain substance to actually be able to go in. So it allows certain substance to actually go in. So here you can see that is an actual channel. So this nuclear pore spans across the whole lipid bilayer. So across this whole lipid bilayer, you have these channels that allow certain things to go in and out. So once the DNA, for example, you take a section of the DNA that you're interested in transcribing, so that is that you want to make, it contains information, let's say, to make a certain protein. So we want to be able to access that information and transcribe it into RNA, which is another nucleic acid, transcribe into RNA, and then that RNA, we can then push that RNA out of its nuclear pore so that it can find ribosome that it can then use to actually translate that RNA into our final polypeptide, hence our protein that we'll need later on. Okay, so similar to the cell membrane, it is a lipid bilayer. So you can see here is a lipid bilayer. We actually have two lipid bilayers. So we have this guy up here. We also have this one right here. So one side will be facing the nucleoplasm, whereas the other side will be facing the cytoplasm inside the cell. So this is our nuclear envelope. Okay, so other Other structural part of the nucleus is the nucleoplasm, which similar to cytoplasm is this viscous liquid right in here. So we can kind of think of it as the purple stuff. And it's similar to cytoplasm. It is, it kind of helps keep support of the different other components that are within the nucleus. We also have the nucleolus, which is this guy right here. This is the dense part in here. And this nucleolus is where we have ribosomes assembling, ribosomes assembling. And ribosomes is made of RNA and proteins. All right, so we'll talk about ribosome a little bit more later on, but just keep in mind that ribosome is what makes protein. So you see this little red dot, that's what I'm pointing to. So these are other ribosomes. Also within the DNA, within the nucleus, of course, is the DNA. So the DNA, we tend to think of DNA looking like this as a chromosome, but a lot of times the DNA will actually look more like this. And so it's not as condensed. It's a little bit more loose, and this is called chromatin. So the chromatin is made of DNA, but it also has certain proteins. And so we can kind of see here that it also has certain proteins that the DNA kind of wrapped around. And so it's more loosely wrapped here and as a chromatin than it is when we form chromosome. But it's loose here, but we still have the DNA wrap because we don't want the DNA to be accessible by the different structures, other structures within the nucleus at any given time. So we want some organization, order, and so the DNA will actually wrap around these proteins, and it's forming what we consider a chromatin. It's the DNA and the proteins. But when we're ready to divide, that's when we need it for the DNA to be a lot more condensed. Because when we want to divide, we actually want the DNA to be dense enough that we can actually organize them and separate them, right? So think of dividing as going from one cell into two cells. So we want to duplicate and kind of organize the DNA so that they line up properly, so we have equal amount of DNA that goes to this cell as this cell. So then right prior to cell division, the DNA is going to compact into a chromosome. And a chromosome will contain one single DNA molecule. So we have, in human cells, we have 46 chromosomes because we get 23 of them from our mom and 23 of them from our dad. So you probably have heard of chromosome 1, 2, X chromosome, Y chromosome. Each one of those is a single DNA molecule, and it's associated with certain type of proteins. Okay, let's move on to the endomembrane system because it's kind of this system is continuous with the nucleus. So the endomembrane system is a series of membrane or cover organelles. So all these organelles have double membranes to them, and we're going from the nucleus to the plasma membrane, which is on the outside of the cell, right? So this includes the nuclear envelope itself, because the nuclear envelope is that double membrane that we're talking about. So we go from the nucleus to the endoplasmic reticulum. So we start off with the rough endoplasmic reticulum, which has, and it's rough because it has all of these different ribosomes attached to it. And these ribosomes will help make proteins. So as the DNA makes RNA and the RNA gets in here, the RNA will actually go to these ribosomes and be translated into protein. And then we move from the rough ribosome, or rough ER to the smooth ER. So this is continuous. You can see right here that is still pretty continuous as we move from rough ER to smooth ER. And so the smooth ER is also part of the endomembrane system. Again, it's also a double membrane. It also has a double membrane, and it's going to be linked to between the rough endoplasmic reticulum and the Golgi apparatus. And so coming back to the smooth ER. While the rough ER is kind of synthesizing protein, the smooth ER is actually doing a lot of the lipid synthesis, and so we're actually able to form like a lot of phospholipids within the Golgi apparatus. Whatever is made here in the endoplasmic reticulum by these ribosomes, the different protein will actually go through the smooth, and in the ER lumen themselves, this protein is undergoing different post-translational modification. So we don't need to know too much, but one of the things, for example, that we mentioned previously when we're looking at the plasma membrane is the glycoprotein. So a lot of times the sugar that's added to protein will happen within the ER lumen. And then, as I mentioned, this endomembrane system continues from the ER, from the endoplasmic reticulum into the Golgi apparatus. And it is within the Golgi apparatus that we're going to modify, we're going to sort, we're going to package the proteins and the lipids. And you can see all the different packaging, we're going to place them in these different packages. So you can see one kind of being formed right here. So we're going to place the protein, the lipid into these packages that would then break off as vesicles. And these vesicles can then be sent off into the cytoplasm for certain for us to move these protein lipids to wherever we need it. But one of the places that it's going to get moved to is actually to the plasma membrane. And this is why we consider plasma membrane as part of the endomembrane system, is that we'll actually move these vesicles into the membrane, and once it's fused with the plasma membrane right here, once it fuses with the plasma membrane, we can actually take the different protein lipids that we made here and secrete it to the outside, so outside of the cells. And then these different molecules, depending on what they are, can actually be transported to other cells. So for example, we might have had another cell over here that made the same thing. It made different vesicle, it went to its plasma membrane and it secreted out. And now these molecules can be taken up by the cell. So it will actually uptake. So these vesicles over here, so let's say this is a vesicle. These vesicles can combine to this plasma membrane, and this plasma membrane can then actually fuse it and then release these molecules into another cell. And so the endoplasmic reticulum, as I mentioned, has two types, right? It has the rough ER and the smooth ER. And we already talked about how the rough ER has the ribosomes on them, and it's the one that can be used to actually translate that RNA into protein. And then within the ER, once it's translated, we can undergo some post-translational modification. We also have the smooth ER, still part of the endomembrane system. Smooth ER does not have the ribosome, so we consider it smooth for that reason. And it is going to synthesize different lipids. It also has other function depending on the cell that it's in. Sorry. So for example, in the liver, we can actually detoxify different drugs within the smooth ER of the liver. And if you're looking at cells that are within the testicles, it can actually synthesize testosterone, right? Which is another type of lipid, right? If you remember, it's a sterile. The lipid is a sterile, and that's the 4 fused carbon ring. We talked about that in chapter 3, but that's a type of lipid that the smooth ER within the testes can form. So this function of what it can make and do, such as detoxing, will vary from cell to cell. But both ERs, both the rough and smooth, can transport the vesicles over to the Golgi, all of that protein and lipid into the Golgi apparatus, which will then break off, and we can send that, again, as we mentioned, to different parts of the cell as well as to the cell membrane. Let's look at the Golgi apparatus in a little more detail. So as we mentioned, this is the transfer station. That is going to help transfer all of the different proteins and lipids that were synthesized. It's going to help that were synthesized in the ER. It's going to help put them into vesicles that we can then package up. So it's going to sort, it's going to package. It's kind of like a delivery system. So once the delivery is made up here, all the different material for that delivery is made in here. In the Golgi apparatus, we're actually going to package it up. We're going to package it. We're going to place them in these vesicles, and then we can send them off to the plasma membrane or to the lysosome. So lysosome is another organelle. Right here, we haven't really talked about that yet. And this lysosome, one of its main things is it contains different type of digestive enzymes. So different enzymes that can kind of play a role with the different content that's in these vesicles. So the different proteins and lipids that are within these vesicles. So once we have sorted, packaged, and stored the different lipids and the different proteins in the Golgi apparatus, we're going to move them to the edge right here, and then we're actually going to form these vesicles, right? So these vesicles are also double membrane, so they're these membrane enclosed sac, and they will actually store whatever substances that we get from the Golgi apparatus, and then they can actually bud off. So you can see here, this budding is kind of happening. So we have the storage right here. It can bud off, and eventually, my pen is off, sorry. Eventually, it can kind of break off and kind of form its own little vesicle. Oh, so we can kind of see that happening here. So we can actually break off, and then you have these vesicles that can then be used to transport things. It can transport things to outside the cell. It can transport things to different parts of the cells. And so one of the places that it can transport is it actually can transport to the lysosome. We won't play the video right now, but you guys can watch it on your own. So lysosome is one of the things that I mentioned. So enzymes that are destined to go to lysosomes are actually made in the Golgi. So enzymes, we recall that enzymes are protein. So these different proteins that are digestive enzymes that we're going to need in a lysosome are actually made in the Golgi apparatus. And so these vesicles, the lysosome vesicles themselves contain digestive molecules. So they have digestive enzyme, and the function of them is basically to break down different cell parts and different waste product of the cells. So similar to how we as a whole organism can make certain waste, the cell can also make waste. And so some of that waste will be transported over to the lysosome so that it can be broken down into smaller products. So here you have the rough ER. It's making whatever proteins that it kind of needs to do, is doing any type of post-translational modification it needs to, including adding, for example, sugar molecules to it, and then it gets moved through the smooth ER into the Golgi apparatus, where it's going to be sorted and packaged. And so let's, for example, say that it's the digestive enzyme. So the digestive enzyme got made here, right, because the enzyme is a protein. And after it's pre-modified, it gets sent to the Golgi apparatus. The Golgi apparatus will place it, make a little vesicle. The vesicle would then, containing the lysosome enzyme, would then bud on to a lysosome itself, deliver that digestive enzyme. The digestive enzyme within the lysosome can then, and the lysosome as a whole, can then bud on to other vesicles, and so then it can digest the content that's inside this vesicle and digest it so that it either removes it as a waste product, or it can break things down from outside the cell into smaller molecules that then the cells can then use. And the last type of vesicle we're going to talk about are vacuoles. And so vacuoles are larger than lysozyme. They're kind of larger vesicles that we can use to store things in there, such as liquid, different type of food content, and then it also can be used to store waste disposal. So we saw the vacuoles when we were talking about them in plants, but you can actually observe some vacuoles in animal cells as well. Okay, so that's it for this section. We'll move on to the next section, which is going to be talking about energy-related organelles.